Golgi operators. Mainly Golgi operators also they contain cisternae. Okay, cisternae vasculature in the Golgi vacuoles. Main role is the role of the secretion, especially in the production of glycoprotein, cellulose, different types of the protein. Next one, mitochondria. Mitochondria is the double membrane model. Double membrane model is also known as the chondriosomes. It was discovered by the Kolliker and Altman in 1880. Later, this name was given by the Benda in 1897. Is the term mitochondria. Generally, these are the rod shape, the diameter of about the 0. micrometer and length of the about 7 micrometer. Each mitochondrion is bounded by two unit membranes. The inner membrane is invaginated into the lumen. Mitochondria from the finger-like projection is also called as cisternae. See the internal structure of the cisternae: matrix, cristae, and ribosomes. Outer membrane. Actually, the space between outer and inner mitochondrial membrane is also called as the perimitochondrial space. That the actually mitochondria is the double membrane model. Double membrane model. The central space of the mitochondria inner mitochondrial membrane filled with the viscous matrix. On the cisternae along with the inner membrane, regularly spaced stock with the particles are present known oxysomes. Is also called as the F1 particles, F1 granules. The elementary is also called as an elementary particles. The matrix contains the proteins, lipids, the oxidative enzymes, coenzymes, and variety of the inclusion like DNA filaments, ribosomes, calcium containing granules. Mitochondria are for the cellular respiration involved in the oxidation of food and synthesis of the ATP. There is therefore is also called as the powerhouse of the cells. Mitochondria is the powerhouse of cells. Next, next one is the mitochondrial double membrane model. Next one is the plastids. These are also the double membrane models. Actually, mitochondria, endoplasmic reticulum, and next one is the chloroplast. Next one is the nucleus. Also, this is the double membrane model. Okay, plastids. The term plastids was coined by the Schimper in 1883. Mainly, these are the commonly they can see into the eukaryotes, especially in plant cells. Maybe the colorless or colored. The colored plastid is also called as the leucoplast. The colored plastid also called as the chromoplast. Colored means chromo. Chromo means colored. Leuco means colorless. Leucoplast is usually have the storage function and the store of the starch and lipids. The green color plast is also called as the chloroplast. The green color plastids are also called as chloroplast. Chloroplast usually the ellipsoidal or discoidal shape. The measure about the 5 to 10 micrometer along with the thicknesses. About the 220 to 50 chloroplasts found in each photosynthetic cell of the leaf. The chloroplast envelope, the chloroplast envelope consists of the two membranes. Each membrane is the reported to be the 6 to 10 nanometer thick. These are the two membranes are the separated by translucent space about the 1 to 20 micrometer wide. The outer membrane generally appears smooth, whereas in the inner membrane is mm, absorbed with the finger-like proportion, like the stroma. The internal structure of the chloroplast is complex membranes running into the matrix. The matrix is also called as the stroma. Mainly, these are the consist of the proteins, the different types of the granules also they are containing the stroma. The membranes inside the chloroplast come into the system. The thin sacs is also called as the lamella. In some regions, several sacs or the lamella are stacked at the top of the each other from the grana. Generally, 4 to 60 grana the found in each chloroplast in photosynthetic cell. Each granum consists of the stacks and the flatted the lamella known as the thylakoids. The stroma contains the ribosomes, circular naked DNA, lipid globules, the starch granules, etc. All the enzymes necessary for the photosynthesis are present in the stroma. In the thylakoid layers, the photosynthetic pigment are present. These pigments are the present in the small groups forming the photosynthesis units. These units are known as the quantasomes. Each quantasome consists of the 3 to 600 of chloroplasts. These are very important. How many number of the chloroplasts they contain the quantasome? 300 to 600 chloroplasts also they contain the each one quantasomes. Carotenoids, phospholipids is also they present in the quantasomes. What are the functions of the in chloroplast? Chloro means color. Leucoplast is the colorless. Okay, chloroplasts participate in the process of the photosynthesis and prepared the carbohydrates. The colorless plastids helps in the storage of the food materials. Leucoplasts serve the storage food materials such as starch, amyloplasts, lipids, elioplasts, and proteins, proteinuplasts. Chloroplasts give the color to the flowers, fruits, etc. Next one is the microbodies. Microbodies are the two types, the peroxisomes and glyoxisomes. Peroxisomes also these are also mainly single membrane model. Peroxisomes present along with the chloroplast, the mesophyll cells of the C3 plants. They contain the enzyme necessary the photorespiration, especially photorespiration mainly they can occur into the chloroplast, mitochondria, and peroxisomes. Main peroxisomes are the detoxification process, especially they can form the hydrogen peroxide. Okay, CPM. These are the single membrane model, peroxisomes also. The glyoxisomes, these are the present in fat rich endosperm cells. They process the enzymes for conversion of the into the carbohydrate. Glyoxolate cycle is also called as the C2 cycle. Mainly glyoxomes also they can participate in the in glyoxolate cycle or C2 cycle. The matrix consists of the enzymes which catalyze the breakdown of the hydrogen peroxide is oxygen. They also possess the catalase enzyme which forms the water molecules. Lysosomes is also called as the suicide bags of the cells. These are the spherical vascule which are the bounded by the single unit membrane. The lysosomes contain the variety of 10 to 12 types of the hydrolytic enzymes. These enzymes work for the efficient in the acidic medium, hence known as the acidic hydrolysis. During the cell death, the released outside results in the death of the cells. In this way, they help in the cell ends the lysosomes known as the suicide bags of the cells. Lysosomes also contain the enzymes in the useful digestion of the lipids and proteins and glycoproteins. Chromosomes. Next one is the lysosomes. Also, lysosomes are spherical. Is also called as the suicide bags of the cells. Microtubules in the microfilaments. The cytoplasm of the plant and animal cells are the preserved the numerous ultrafine tubules in the tubulin. The protein called is the microtubules. Microtubules are the complex structure generally composed of the 13 individual proteofilaments arranged in the form of the hollow cylinder. The functions of the microtubules is the formation of spindle fibers during the cell division. They, from the structural unit of the centrioles, vessel granules, cilia, and the flagella, they can composed by the microbodies and the microfilaments. Microfilaments are the thin fibers that are the narrow than the microtubules. They are found in the either freely in the cytoplasm or anchored the proteins. They provide the stiffness, shape, and the cell movement of the microtubules. Is microtubules also consist of the tubulin protein. This is one type of the protein, tubulin proteins. Vacuoles, cytoplasm of the many plant and some of the animal cells also contain the numerous small or the large sized hollow liquid filled the structure called vacuoles. Especially vacuoles also only they can see into the in plant cells. There is no see into the animal cells. Okay. The vacuoles are the bounded by the single membrane, the semi-permeable membrane is also called as the tonoplast. They are the filled with the fluid and this is also called as the cell sap. The cell sap contains the high concentration of the salts, sugars, gases, and the next one is the structure and functions of the cell membrane. Cell membrane also, the cell wall surrounding by the cytoplasm. Okay, structure and functions of the cell membrane. The inner cell wall and the surrounding the cytoplasm delicate the flexible cell membrane, mainly is also called as the cell membrane. Outer structure, the inner of the cell wall, these are surrounded by the cytoplasm. The thin, delicate, flexible cell membrane, the present is also called as the plasma membrane or plasma lamella. Plasma membrane is mainly composed of the lipoproteins. The ratio of the protein and lipid varies, and the ratio depends on the type of the membrane, cell and type of the organism. In addition to the lipoprotein, polysaccharides also seen into the in certain cases. All these chemical components they held together the thin film like the playable sheet of about the 75 angstrom thicknesses. Lipids in plasma membrane, lipids are the present in the form of the phospholipids. Glycolipids. Each phospholipid is the molecule that consists of the head and the tail region. The phospholipids are the bipolar nature, consisting of the hydrophilic and the hydrophobic region. These types of the molecules are known as the amphipathic molecules. There are the round five types of the phospholipids, of the which important in the theotin, choline, cephalonate. The glycolipids are the process the glucose, the fatty acids, phenoglycine. The common lipids also animal cells, the cholesterol and phytosterol in the plant cells. The cholesterol in the animal cells, the phytosterol also are containing the in plant cells. Proteins, plasma membranes consist of the around the 60 to 70% of the protein. There are the three types of the proteins in the plasma membrane. Structural proteins, lipids, proteins, and structural proteins. They're also backbone of the plasma membrane and lipophilic in nature. The next one, functional proteins like the enzymes. These are the participate in the biological functions. Carrier proteins. These are also integral proteins of the plasma membrane. It helps the transport and products of the across the membrane. Basically, the proteins plasma membrane divided into two types: peripheral proteins, and next one is also called as extrinsic proteins. Integral proteins are intrinsic proteins. Peripheral proteins are the present superficially around the two lipid bilayer, whereas the integral proteins with their the hydrophobic ends in the membrane in the plasma membrane. Other constant, some membranes is the in addition to lipids and the proteins also possess few polysaccharides, carbohydrates are also present on the superficial to the cell membrane, forming the glycols, alter structure of the plasma membrane. These are the several biologists have the proved the different models of the plasma membrane models. Bilayer model, the lipid bilayer model also sandwich model, Robertson model, and unit membrane model, Daniel Davidson model, and fluid mosaic. These are the bilayer models. Daniel Davidson model also, fluid mosaic model, Robertson model, membrane, unit membrane models, and micellar model. Among all the models are the fluid mosaic model is the very widely acceptable model, the fluid mosaic model. Fluid mosaic model also proposed by the S.J. Singer and J.L. Nicholson in 1972. According to this model, proteins are the dispersed within the bimolecular lipid layer in the form of the mosaic. Mosaic means the zigzag manner. Zigzag manner. This is just like the proteins are icebergs floating in a lipid sea. In this, proteins are the arranged in the two different types. Some proteins are the present in some proteins are the present on the surface of the membrane, and these are the referred to extrinsic proteins. Some proteins are the present embedded in the lipid layer, and they refer to the intrinsic proteins. The lipid and intrinsic proteins are the amphipathic in nature. These molecules contain hydrophobic properties. Lipids are the organized in such as the way the heads are the faced towards the outside of the membrane and the tails towards their inner side of the membranes. The spherical proteins are the loosely attached to the lipid molecules. The lipids and the peripheral proteins can move freely in the plasma membrane. It then indicates the semi-permeable nature of the plasma membrane. The fluid mosaic model is the membrane, the view dynamic structure with the permeability barrier also serving the solvent and the integral proteins and lipids. Lipids and integral proteins in the bimolecular layer are free of the move laterally within the liquid solvent. The carbohydrates, the motifs, the glycolipids, and the glycoproteins located on the outside of the membrane are believed to be the important in assuring the proper orientation of the proteins in a membrane structure. They also have the crucial role in the phenomena the cell recognition and the interaction. What are the functions of the cell membrane or plasma membrane? Plasma membrane divided into the cell into the small specific functions. Units are the components. This develop into the autonomous bodies called the cell organelles. Plasma membrane is the differently permeable membrane and it is the regulates the movements of the substances in both ways. It permits the entry of the some salt and water into the cell from outside and generally prevents the substances from moving out of the cells. It helps It helps the movement of the particle across the membrane, either the positive diffusion or active diffusion. Protein molecules on outer surface of the membrane act as the receptor which unit with the Complementary substances are the ligands provide the external stimulate of the cells. Membrane provides the structural framework for the location of the various enzymes, and the proteins membrane may act as the carriers of the transport and materials. This is the plasma membrane or cell. And the next topic is the nucleus. A nucleus the nucleus is discovered by the Robert Brown in 1831. It is the control of the all metabolic pathways, heredity activities of the cell. The nucleus only found in the eukaryote cells, whereas the prokaryote does not show the nucleus. The well differentiated nucleus also, especially they can see into the in eukaryotes. There is no differentiated nucleus also they cannot see into the in prokaryotes. Okay, ultrastructure of the nucleus. The consist of the some of the major parts: nuclear membrane, nuclear sap, chromatin fibers, and nucleus. Nuclear membrane. The nucleus is the surrounded by the envelope two unit membranes. The two unit membranes separated by the gap 100 to 150 angstroms. This is the intermembranous space is the known as the perinuclear space. The outer space of the outer membrane is the rough known due to the presence of the ribosomes, and in the certain places continues with the endoplasmic reticulum. The envelope has the several nuclear pores, and the diameter each pore ranges between the 10 to 40 nanometer. Nuclear sap. The interior of the nucleus is filled with the transparent and the granular and the acidophilic fluid called is the nucleoplasm or nuclear sap. Carrier in this numerous proteins, the RNA, hydrolytic enzymes, phospholipids are the present. It also possesses the various enzymes necessary for the mechanism of the DNA replication, transcription, spindle fibers formation, chromatin fibers, numerous threads like the elongated structure of the suspended throughout the nucleoplasm called the chromatin fibers. During the cell division, chromatin fibers are arranged into the chromosomes. Chemically, chromatin consists of DNA and proteins. The proteins are the two types, histone and non-histone proteins. Histone proteins are the small basic proteins, such as the arginine, lysine, especially these are say also into the eukaryotes. Non-histone proteins also RNA polymerase. These are also they can sit especially in prokaryotes. Nucleolus, in nucleolus is the nucleus. Nucleus is consists spherical prominent ball-like body known as the nucleolus. The generally one nucleus is present in the each nucleus, but the rarely more than one also observed, and the nucleolus is attached to the particular specific position on the chromosomes known as the nuclear organizer. There is no separate membrane around the nucleus, and this is the plots in the nucleus sap. Ribosome synthesis takes place in the nucleus and reaches the cytoplasm through the nuclear pores. Nucleolus is the disappears in the prophase and prepare the telophase of the cell membrane, cell division. Nucleus morphology, number and size. Nucleus is present in the eukaryote cells. It is absent in the several elements of the phloem. And also is the mammalian red blood cells also absent, also present. Generally in the young cells of the nucleus present in the center of the cell, but in the matured cell due to the development of the vacuoles is the pushed to side in the cell. Number of generally one nucleus present on one cell in the mononucleate. The tapetal tissue of the latex cell possesses the many nuclei, is also called as the multinucleated shape. The shape of the nucleus depends on the type of the cell. The nucleus is around The nucleus is around 3 to 25 micro radius different organism based on the number of chromosomes and types of the cells. Number of chromosomes. Chromatin substance in the nucleus forms the chromosomes during the cell division stage. If one set of the chromosome is present is known as the haploid, and two sets of the chromosome then is known as the diploid condition. Chromosomes controlling the somatic characters is known as the autosomes and somatic chromosomes. The chromosomes which that determine the sex and organism is also or male or female is known as the sex chromosomes or allosomes. What are the functions of the nucleus? It is undergoes the division during the cell division. The mitotic cell division it helps the undergo the growth and development, and with the mitotic cell division it helps the formation of the gametes required for the sexual reproduction. The chromosomal number of gametes is applied. It plays an important role in the heredity as consists of the DNA. It is the chromosomes through the gene present on the DNA. The characters are the process from the generation to the next generation. Nucleus present in the nucleus plays an important role in the synthesis of the ribosomes. In the nuclear cell sap, DNA replication and RNA transcription takes place in nucleus. The nucleus controls the metabolic activities of the cell. These are the nucleus. Next one is the nucleic acids. Next one is the nucleic acid. These are the two types of the nucleic acid. One is the deoxyribonucleic acid. Another is the ribonucleic acid. What are the major differences between the deoxyribonucleic acid and next one is the uh, ribonucleic acid? Deoxyribonucleic acid is the double standard. Ribonucleic acid is the single standard. At the same time, DNA they can see into the all cells of the organisms. The nucleic, especially ribonucleic acid also they can see into the only new cellus. Uh, Next one is the ribosomes. Is also only present in the and next one is the in nitrogen bases in DNA they are containing the nitrogen base also adenine, guanine, cytosine, and thymine. Instead of thymine, in uracil are present into the in RNA. Especially DNA also they can divide the zDNA, aDNA, bDNA. Okay, in RNA also tRNA, mRNA, rRNA. Okay, these are the mainly the chemical composition of the nucleic acid also. The special nucleic acid is the composed types of the substance phosphoric acid, the pentose sugars, and the organic bases also nitrogen bases. Next one, nucleoside. What is the meaning of the nucleoside? Especially phosphate and the nitrogen bases and pentose sugars also known as nucleosides. There is no phosphate. Sorry, there is no phosphate. Sugars plus nitrogen bases equal to nucleoside. Sugars plus pentose sugar plus phosphate is also called as nucleotide. Nucleotide. How many nucleotides? Especially nucleic acids also DNA and RNA. Okay, nucleotides and nucleosides. The structure of DNA is the primary, the Crick, Watson and Crick model, and the Watson Crick model. Especially these are the Watson Crick model developed by the Watson and Crick. DNA based the chemical and physical biological properties of the DNA. This model is accepted by around the world. Both of the is got the Nobel Prize. Crick model DNA consists of the following different characters. DNA molecules made of the two polynucleotide strands chain. The two strands are the coiled together from the DNA double helix. The two strands of the DNA molecules oriented the anti-parallel to each other. The one strand runs the 5 to 3 prime direction, while the other runs is the 3 to 5 prime direction. Each polynucleotide composes the many nucleotides joined with the phosphodiester linkage between the sugar and phosphate residues. The two acts of the backbone of the DNA. Nitrogen base is the organized in the stepwise manner for parallel each to they are the connecting to the both of the strands of DNA. In each step of the pair, nitrogen bases are present in the adenine. They always bound with the A double bond T, C double bond A double bond T, and G C double bond G is the triple bond. In between the three hydrogen bonds are available. A double bond T is the in two hydrogen bonds are present. Mainly adenine and purine as the both are the they can attach to the Ag is the adenine, purines. The pyrimids are the CT. Okay, these are the CT. Okay, the formation of hydrogen bonds between the AT and GC also known as the complementary base pairing. The hydrogen bond is relatively weak bond, and this is the since in the DNA molecules the several hydrogen bond. The two strands of the DNA molecules are coiled together the right-handed helix forming to the DNA double helix. The results of the formation major and the minor grow in the DNA proteins interact with the DNA and this sites the growth. The diameter of this helix 20 angstrom, while the pitch is 34 angstrom. Each turns the 10 and equally spaced base pairs. The distance between the successive base pairs the 3.4 angstrom. Based on the arrangement of the nitrogen bases, one strand and the sequences, the base of the other strand is the determined. For example, one strand of the DNA as cat, the genetic information present in each strand of the DNA, the strands is known as the template strands. During the DNA replication, the two strands of the DNA separates when the new complementary DNA strand synthesizes. DNA replication is the three types: dispersive replication, conservative replication, and semi-conservation. What is the new replication of the DNA? Is the identical copies. They can form to the identical copies of the DNA. Okay. Mainly semi-conservation model DNA replication also acceptable model. The mechanism of the semi-conservative method DNA replication initiation, elongation, and next one is the termination. Initiation. They can start with the initiation. Okay, the begin the certain unique fixed points is also called as the origin or ORA regions. Initiation is the begins with the uncoiling double strands, two enzymes, the DNA gyrase and DNA ligase, helicase, bind with the origin points and they induce the unwinding, the separation of the complementary DNA. The separation of results Y-shaped fork is the origin, referred to the replication forks, the two strands of the base of the exposed DNA. Elongation mainly is that the catalyzed by the enzyme DNA polymerase. The DNA polymerase can extend the new chains of the DNA in the presence of the RNA primers. And the polymerase primers enzymes generates the next one termination. The termination is the end part. Mainly DNA polymerase enzymes involved in the DNA replication. DNA polymerase 1, polymerase 2, polymerase 3. Polymerase 1 and polymerase 2 and polymerase 3. Types of the RNA, mRNA, tRNA, or rRNA. These are the nucleic acids. Thank you.